Good morning. My name is Bill Nelson, and I'm honored to be here today to talk with all of you about the impact of immunotherapy research and just how the V Foundation is contributing to progress made in this field. Joining me for this discussion are two incredibly brilliant characters. First, we have Dr. Craig Slingluff, the Joseph Helms Farrell Professor of Surgery at UVA Health, the Vice Chair for Research in the Department of Surgery, the Director of the UVA Cancer Center Human Immunotherapy Center, and co-chair of the Melanoma Committee of the Eastern Cooperative Oncology Group. Also joining us today is Dr. John McCarty, the Medical Director of the Cellular Immunotherapies and Transplant Program at the VCU Massey Cancer Center. Thanks to both of you for joining us today, and thank you to each of your respective institutions for being the mission partner sponsors of this, our Virginia Vine event.
Bill, thank you for the offer. Thank you for giving the opportunity.
So we're here today, John and Craig, to talk about the immune system and how we're increasingly use it in the battle against cancer in individual patients. So the immune system is so remarkable. It's capable of distinguishing an infected cell from a non-infected cell, your cell from my cells, friend from foe, all this kind of stuff, even cells infected with SARS-CoV-2, the COVID-19 epidemic versus non-infected cells. Can the immune system Craig, can the immune system see cancer cells differently than normal cells? And if so, how does it do that?
Bill, thanks for asking. It's a great question. And as you well know, this has been a debate over a period of decades. But there's data from a few decades ago that's quite convincing that if you just do some changes to a mouse so that its immune system doesn't work as well, it will spontaneously develop cancers that it wouldn't otherwise get. And we also know that patients who are immunosuppressed can get more cancers. So it's clear that there is some role of the immune system in preventing cancer. We now know a lot about the target molecules on the surface of melanoma cells and other cancer cells that the immune system can recognize. And we know that these are the mechanisms that distinguish the cancer cell from a normal cell. But there's also, an important factor in how the immune system recognizes its targets. Partly it can recognize self and non-self, but it also recognizes danger. So if cancer cells are causing damage when they're arising, they may cause the immune system to be activated.
So John, talk to us about when you think of the immune system, it's not one cell, it's a whole collection of different cells, many of them with very specialized functions. They're T cells, which are we think of as the smart cells that can recognize components of the insides of cells presented at the surface, as Craig was talking about. But there are B cells that can grow into cells that make antibodies, which are sort of smart bombs. There are, and the antibodies are the kind of things that we try to elicit by vaccination to combat SARS-CoV-2 and the COVID-19 vaccines. There are macrophages and other cells that just eat stuff. How does what do each of the components do, and are each of them being targeted to help with cancer?
So that's an excellent question. And yes, the immune system is highly complex, and we continue to learn more and more about how we can both adapt it, use it, and make it more effective in its job. There's adaptive immunity, there's adoptive immunity. What are those things? Well, there are cells that recognize that danger, recognize inflammation, recognize injury, that essentially are to address those and clean up any injury that may occur. But we also need the ability to respond to new threats. So COVID-19 is a completely new antigen. Not long ago, we dealt with H1N1. Those are viruses, perhaps, that produce new proteins on the cell surface that the immune system may not have seen before. And so there needs to be the ability of the immune system to adapt to new challenges, recognizing something that is not normal, not self, and of a threat and a danger. Eventually, these cells will not just do their job and disappear, but they'll actually be permanent or semi-permanent sentinels to continue to watch for these challenges to come up again to respond effectively. And that's where we think about vaccines having a duration or a durability over time.
So Craig, John mentioned that the immune system is very well constructed to recognize new threats and build responses to them. One of the things that all cancers are fundamentally disorders of acquired defects in genes. And the genes, of course, are the blueprints for all the components of the cancer cells. And so these components are new things, part of new threats. And the immune system can see these defective genes, the mutations, if you will, as they play out. How do they see those?
Well, they can see them by different mechanisms and by some of the things that John's already mentioned as well. So antibodies can see them if they're on the cell surface, if it's a molecule that's altered on the cell surface, and T cells, as you've mentioned, can see them if they're changing proteins inside the cell, but fragments of those proteins get put out on the cell surface. I always like to tell my students it's the immune system sort of works like the detectives you see in some TV shows where they don't know what's going on in some suspect's house, so they want to look in the trash to see what he threw away. And so the cells do the same thing, whether it's virally infected cells or cancer cells. They'll put portions of the proteins that are altered out on the cell surface like trash, and the immune system cells can see that and then target the cell.
Oh, I love that. I love that analogy. So here's one I'm going to ask both of you, actually. Maybe John first. So if the immune system can see cancer cells as distinct from normal by looking at the trash that they're holding up on the outside, why doesn't the immune system eliminate all cancers? Why do we get cancer in the first place? If it can see it, what's holding it at bay? Why do they grow to threaten life in the first place?
Yeah, that's the question that we're all trying to decide, and I don't think there's one specific answer for that. And actually, I would even argue that it can't always see. The prime example is a certain kind of lymphoma, B-cell lymphoma, where very often the cancer itself looks too close or too much like a normal lymph node or a normal lymph node cell that the immune system can't recognize it. And that often happens where there are no cells that recognize the cancer as being foreign. Sometimes there are those kinds of cells, those immune system cells, but they're not there in present enough quantities, or they're not mobilizing quickly enough given the speed at which the cancer may be growing and developing in some aggressive situations. And then the other is that both the environment which the cancer is developing, or even the cancer cells themselves, can essentially put the immune system that is attacking it to sleep. And that's where we'll talk a little bit later about some of the addressing all these things, how we address with cellular therapy and with some of the checkpoint inhibitors to cover many of these main mechanisms. I'm sure there's certainly more, but those are the main ones.
So Craig, John described sort of at least one mechanism in addition to the going to sleep on, which we are going to talk a little bit about. But
yes,
but this is the stealth notion that present the antigens or you're not that different. You're an expert in melanoma. Melanomas often arise as a result of a barrage of things that cause mutations. They often have a very large number of defective genes, a large number of mutations. Very interesting trash. The world indoor record may well be syndromes where they just acquire mutations all over the place, so-called mismatch repair defects. But so what do you think's in play in melanoma? I guess you could argue that there was some evidence all along. It's one of the reasons people like you are in this game. There's some evidence all along that the immune system occasionally seemed to respond to melanoma, didn't it?
That's right. Yes. So there definitely have been examples like that, and there was a book published in the late 60s that summarized spontaneous regressions of cancer that had been documented in the literature, and melanoma was one of the most common where that happened. There's a patient that I know was treated here at UVA many years ago, and I wasn't directly involved in his care, but learned later, who came in with a melanoma on his forehead. He was an older guy, and he didn't want to go through too much stuff. They biopsied it, showed it was melanoma, and he didn't want to have it taken out. And he had something in his lung that was questionable, but they thought it wasn't bad enough to do something about. He came back later, the lung things had grown, they found out that was melanoma, but when he came back for that, they saw the thing on his forehead gone away, all by itself. So there's a concept that Bob Schreiber has talked about called immune equilibrium, where, and I think this is a big part of what goes on, there's a constant interaction between the immune system and cancer in those cases where the cancer really is being seen by the immune system well. And sometimes the immune system can win that battle, but a lot of times the immune system kills some cells, and the cancer cells figure out ways that they can escape immune recognition, and they grow, and then the immune system adapts, and then the cancer adapts. And if you'll forgive me making another analogy, I used to wrestle in high school, and my coach would tell us the way to do a takedown, to do something, and then we'd think, oh gosh, we're powerful now, and then you try it and he'd show you the counter, the way to prevent that from working, and then you think, now I have no power at all. But the line he would say is, there's always a counter to every counter. So the goal of the immune system is to overcome whatever the cancer does to counter its effects.
So I'll stay with you, Craig, on this one. And so in the, you have this, some people call immunoediting, I guess, right? You have this interplay between what the immune system is recognizing as the cancer develops and what sort of sneaks through in the end if it's growing to cause symptoms or threaten life. What we've learned many times is this business, as John was talking about, is sort of putting the immune system to sleep, or the metaphor, I have parented teenage daughters. I always viewed it as the talk to the hand response, that you have this scenario where smart cells in the immune system have shown up, they've muscled up in great numbers, they're right there where the cancer is, but the cancer is giving them talk to the hand, we're holding you in check. And holding the immune system in check is something that, as we've understood this a little bit better, has led to huge advances. So what is this notion? What is an immune checkpoint, and how do these inhibitors disable it?
Yeah, it's been a big deal. And for those who haven't kept up with the developments in this, the discoveries of these immune checkpoints led to the Nobel Prize in Medicine 2 or 3 years ago, and has revolutionized the treatment of many cancers. The one thing to think about is that the immune system works beautifully. It's an amazing symphony of cells and other molecules interacting, but like any system, it doesn't always work just right. And so on one hand, it can fail to recognize cancer. On the other hand, it can wrongly recognize normal tissue as something it should destroy. And that's what leads to autoimmune diseases like Crohn's disease, ulcerative colitis, diabetes, other things. So there are mechanisms though to try to make that happen as little as possible. And so it makes sense that the immune system has brakes on it, so that the tissues that are normal will do things that prevent the immune system from recognizing it. And one of my colleagues has pointed out once that although most of us think about these immune cells as being the thing you're looking for, the thing you're trying to have lots of, the reality is normal tissues that are doing their job don't want the immune cells there. Just like you don't want police with arms, with guns walking around your neighborhood. You kind of want things to be peaceful. And so there are mechanisms that say we're going to stop this immune response. And so that's good in normal tissue, but what happens is cancers express these same molecules that block the immune system from functioning in normal tissue, and then can turn them off.
And these immune checkpoint inhibitors have pretty much revolutionized the treatment of melanoma, the disease you know so well. And as you did in your comments, the side effects that we look for very different than chemotherapy side effects, aren't they?
Yes, they are.
These autoimmunity kind of thing, just what you were saying, right?
Exactly, right. You're sort of shifting the balance. And obviously, if you've got somebody who's dying of cancer, where the immune system has not succeeded in controlling it on its own, we're willing, most patients and physicians are willing to take a risk of some autoimmune side effects for the sake of destroying the cancer. And because they work really well and for a long time in many patients, these molecules, they're antibodies that block these checkpoints, are wonderful drugs. But then there's a whole new body of study that our medical oncology colleagues spend a lot of time on, understanding how to treat those. Unfortunately, a lot of those treatments can work well to prevent those side effects.
So John, in your comments earlier, you foreshadowed something incredibly well. You talked about how, for B-cell lymphomas and other things, perhaps the T cells aren't easily recognizing things. There's another solution that the world has conjured up for that problem, which is to build a better T cell. Take the T cells out of the body and build a better one. How does this work?
Sure. Well, actually, I'm going to even step a little bit further back. We've been doing a fair amount of cellular immune therapy for a while. think about, in addition to the checkpoint inhibitors, which are relatively new, allogeneic transplant, where basically we bring someone else's immune system in, a donor that matches. We're really not just trying to find a clean bone marrow that doesn't have the cancer. We're actually giving that person a new immune system that is going to be less tolerant of having that cancer the patient has, lying around. It's almost like a person moving into a new house saying, I like the house, but I'm going to redecorate it in my way because I will not tolerate having that cancer in the place that I live. But it also, like the checkpoint inhibitors, remains nonspecific. And so there are autoimmune complications that can be very dramatic and can be very problematic for patients. So it gets into what you're talking about. Right, exactly. Graft versus host disease, which is certainly a big problem and can be a problem for patients for years. So how can we find the best of both worlds? And that's where other forms of cellular immune therapy can work. And so you alluded to CAR T cells, or chimeric antigen receptor T cells. Here, we take T cells that might already have a day job. Maybe they're supposed to remember the last flu shot you had, or perhaps that pneumonia that you had a while ago, or that colon cancer that it successfully held off. And we take them and we now give them a new job. So remember, we talked about cells that could not recognize the immune system. Now we're actually going to give them genetically reprogrammed, download a new app to those T cells, and now say, you are going to go after this protein. You are now going to go after this cancer. Using Craig's analogy of the police, this is kind of like policemen who have a photo of a miscreant that they have to go look for, and they are walking their beat. They're going around the town looking for that particular person. And when they see it, then they're able to arrest, or in the case of CAR T, attack it like a normal T cell would. There are also other versions of cell therapy, like T cell receptor therapy. These are similar to CAR T in that they also are engineered and excited, but they're almost cops that are allowed to go inside a building. They're not. They can get off the street. They can go in the building and look for bad things going on and arrest people. And then TIL cell therapy, which are tumor infiltrating lymphocytes, the proof in principle was with melanoma, as you know, Craig. And I liken those, if we're really going to beat this analogy to the ground, as undercover cops that have recognized badness and bad behavior. And so we can take those and expand them and give them the stimulus. So now not only are they recognizing the cancer, they're now stimulated and expanded to be enough numbers, and that hopefully that expansion and that excitation may overcome that talk to the hand problem that we've alluded to before.
So John, as we're reimagining the police as the immune system, we're going too far with this metaphor, perhaps, but it is fun. One of the things that Craig will tell you, with the immune checkpoint inhibitors and melanoma, some of these responses are just plain breathtaking. They are for non-small cell lung cancer.
Absolutely.
Just things I think we all have to argue. We just haven't seen something that's promising and that exciting before. This is true for some of these CAR T cell responses as well for children with acute lymphoblastic leukemia. And so, talk to us about some of those. Have you seen some remarkable things?
Absolutely. In fact, the very first patient that we treated at VCU with CAR T for diffuse large cell lymphoma, this gentleman was actually having a very accelerated immune response just from his lymphoma alone. Fevers, night sweats, couldn't eat, had really no appetite, was losing weight, just very, very, very ill. And when we did some of his imaging, his PET scan, it was very clear that not only all the lymph nodes in his abdomen were very heavily involved, but his liver and his spleen were highly involved with lymphoma, and were very, very bright and very positive on the testing we did. We gave him the CAR T cells, and within the 30-day window that after the infusion, his PET scan, his scans all went to negative. He got a complete remission, a complete disappearance of any evidence that we have for his lymphoma. And he did very well with it, with very few complications, fortunately. Actually, his late complication is that he drank too much Red Bull one day and became very manic, but because we were watching him so closely, we admitted to make sure everything was okay. But basically, had, a incredible response.
So Craig and John, I'll ask you both to do a little bit of future leaning. So the environment around the cancer, in technology being applied to study this is astounding as well. They're really looking at what each and every cell in there may be doing, what its agenda is, what it's up to. And to me, it's easier to use an ecology metaphor. The cancer cells are clearly corrupting this environment. They're polluters and every imaginable thing.
Yep.
But what it seems to be happening is that in that environment, for many different mechanisms, holds the immune system in check. But what that means is there appear to be many different targets that we may be able to aim at to bring the immune system into play when we want it to chase after cancer. I'll just ask each one of you, Craig, what's your favorite target you see on the horizon that may add to these incredible responses?
Yeah, it's a great question, and it's hard to narrow it down to just one because, as you say, there are lots of processes we try to address. There is certainly, well, I do a lot of work on cancer vaccines, so a lot of it's trying to activate the immune system in the ways we've talked about, but we need to do something more than that. So adding molecules that help to support that immune response, so co-stimulatory molecules, antibodies to support them is good, but also directly changing the tumor microenvironment with tumoral injections of immune activators can be very effective, and there's certain approaches like that are approved or showing promise already. So I think that's a great way to go. We do some work here with focused ultrasound, attempts to locally change the tumor microenvironment, sort of reset it and help with antigen presentation. But there are many, and I think this is where there's such a need for more research that can study each of a number of different potential targets and to understand how they speak to each other and interact with each other to help treat people that we can't otherwise treat effectively.
So John, Craig sort of also hinting that this appears to be wide open for opportunity. What do you see as the next things on the horizon?
Well, I think it's not so much looking for a target, a specific target. I think it's actually an approach. I mean, many of us have been doing this long enough. I remember in the 70s, we treated Hodgkin's disease with essentially one chemotherapy. And subsequently, we started treating with multiple chemotherapies because we knew that each had a different mechanism of action. And sometimes it's better to attack a problem from all sides. We did a sort of a combination where we started adding antibodies and other immune-related agents to chemotherapy to further attack the problem in a different way. And I think that's the same thing here. And the difference is not just adding one CAR T cell that looks at one protein on the lymphoma surface and then see if that did its thing. It's going to be looking at how we potentially attack multiple targets at the same time, or as that lymphoma is injured by that initial CAR T attack, releasing new potential targets that can basically have follow-on attack. Combining some of the checkpoint inhibitors with CAR T, trying to find that balance between a very powerful and durable response, but also one that doesn't increase the side effects and the toxicity that can occur from a very accelerated immune response as well. I think the bottom line is it's learning more about biology. It's learning the biology of the immune system. It's learning the biology of the various cancers for which we don't have effective therapies, or even ones we do, to be more effective. And then learning more about the interaction when these new therapies interact. That wrestling analogy of how do we continue to create new counterpunches? And maybe we have to be a little unfair. If you've got a really good wrestler in the ring, maybe we got to get the whole team on, inside and try to take that person down instead of just one-on-one. I think that's where our direction is. And often that's the least sexy part of it, right? the thought of supporting and investing and funding a multi-center grant for a new drug that is going to revolutionize the treatment of sarcoma is very exciting. But what got us there is that basic science, understanding the biology, understanding how we can find new targets. That was the key component, the first start of getting us to where now we're starting to do these treatments.
And so Craig, we sort of, got you to look into the future for where the research may be taking us. How about where the impact of all this on clinical care is? I mean, you're a surgeon. Cancer surgery is completely different than it was when I started. Surgeons operating through little holes using telescopes and all this kind of stuff. What's this going to mean for cancer care? How's this going to impact a regular person with cancer?
Right. It's a great question. And certainly we're already seeing that some scenarios where we would say surgery is our only option, and maybe not a great option, but it's what we'll try to do, we don't do anymore. And instead we'll try systemic therapies with the checkpoint agents or others that can really have a great effect for a lot of people. But and so that has replaced some surgical management. On the other hand, it's fascinating that we sometimes see a situation where someone may have many tumors and most of them respond to the therapy, but one or several that we can cut out continues to grow or causes some symptoms. And then we can go in and take those out. And it used to be we wouldn't really think that was a meaningful option, but I think we're seeing more and more cases where the immune system is working kind of locally in different microenvironments within the patient, and where it fails, surgery can have a role. So there's a continued evolution in how we apply the tools we have, including the expertise of different specialties in the treatment of cancer.
And John, I'll ask you a similar question. You mentioned Hodgkin's disease. The evolution of treatment in Hodgkin's disease is fascinating. Remember, we used to chase after with radiation and chemotherapy and all this kind of stuff. And as we built better systemic therapies, the need for radiation sort of waned a bit, and we could get pretty far along with chemotherapy, and slowly but surely even a little bit less noxious chemotherapy, we hope, in the future. Is that where this is headed? Get systemic treatments that don't bang up people so much?
One would hope so. I mean, ironically, we're even seeing that there are patients who are receiving the CAR T therapy, for example, who might otherwise, partly because of their disease, not be eligible even for an autologous transplant where they serve as their own donor because of perhaps prior chemotherapies and so on. I think what we're going to see is that these cellular therapies, things that are being positioned and approved as you get this when you've been resistant to X number of different cycles of chemotherapy, to start moving them up earlier and earlier and earlier in the treatment process, in the phases. One of the issues that we have is that when you start to take immune system cells from a person who's been through multiple rounds of chemotherapy, that immune system has kind of seen a lot of road. And so trying to get immune system cells earlier when they may be more robust, maybe more facile, maybe more agile, then many of these therapies may be more effective and perhaps with less toxicity in that way. I think precision, this is where the tie into precision medicine also takes place, because perhaps the ultimate thing is to sequence someone's cancer, find out what the unique proteins or what might be uniquely expressed in that person's unique cancer, create a cellular therapy or a targeted immune attack against it, and really get it down to precise laser-focused therapy. Love to see that in my lifetime.
Well, and this is clearly keeping up with this thing that we've learned so well by doing all the sequencing of the cancers, the DNA sequencing of the cancers, and that every individual is unique. The cancers that appear in the different people are similar at sometimes, but actually quite different from each other, and an increasing opportunity to individual-ized treatment. Personalized, you could say, you could argue all cancer treatment is personal. So individualized cancer treatment, and working with the immune system is going to add to that, not undermine it at all. So John and Craig, the V Foundation has funded a lot of research in cancer immunotherapy. And from the discussion we're having today, you can clearly see why. What's your impression of this investment, or the reason we need more of this investment now in this kind of research?
I think what I see is that you really need to invest in every phase of the research. Kind of alluded to that earlier, where sure, I would love to have a great idea, walk up to the NIH or to a very large pharma company and say, make my great idea happen, but it isn't how it works. In order to take a great idea, you need to have and parlay it into a study, parlay it into funding, parlay it into a grant. You need to have data, but in order to get data, you need to have a grant. So very often we're in a big catch-22 about how to make those first steps. Plus, there are many other steps leading up to where you can actually write a multi-center or an NIH type grant. And that's where funding and support from places like the V Foundation very specifically, has helped launch that, created beautiful soil in which we can put the great seeds of ideas and watch them grow into something much more effective and something more long-term. That's essentially how CAR T cells started. They started in laboratories, either at the NIH or at universities, and have expanded from there. And we recognize them now as pharmacology agents, but that isn't how they started. It's an understanding in that basic biology and that basic means by which the immune system works. Not very sexy because it doesn't have a drug, it doesn't have a label to it. But that's how we got here. That was the essential and incredible essential starting point.
So Craig, the V Foundation has supported translational research in cancer immunotherapy and other aspects of cancer treatment. But the other thing that they've invested in is the human capital that makes the big difference, is the V Scholar program. You were once a young surgeon thinking about what you wanted to do, how you wanted to change the world. Talk about what it means to be a young researcher, the kind that's supported by the V Scholars program.
It's a really important point and question. There are two reasons it's important. One is for the individual who's a young investigator. Often the challenge of getting funding that can help you maintain a career and do good work for decades is hard to get. And so getting some initial support to develop your career and to develop your expertise at a young age will help you be successful long term. And then also for the field, whether for cancer therapy in general and immunotherapy in particular, we need more and more people to do this kind of work. So if we don't invest in bringing up people into this field, we're not going to have them in the future and for the long term. So it's really, really, really key and really important.
So the V Foundation is really well poised to shape the future for this cancer immunotherapy and ultimately getting a cure for cancer.
The distance between the bench and the bedside in oncology and in hematology is very, very short. And so immunotherapy is a prime example about how making those steps effective and funded have resulted in very meaningful things for our patients.
Yes, and
so
we'll
thank you
continue
for what
to.
you do.
Wow, this has been an incredibly invigorating conversation and a glimpse into the future of cancer immunotherapy and cancer research, just the kind of thing the V Foundation has been supporting in a way that's going to change cancer. It's going to cure cancer ultimately. And with that, we're going to turn this back over to our friends Jay Billis, Ralph Sampson, and Derek Wittenberg over at the Salamander Resort and Spa in Middleburg, Virginia, who are going to take us to the next part of the program. Thank you, Craig and John, and take it away, Jay.